All right, get going here. So again, these are genes that go up and down. What we can also do, and what's more important with therapies, right, is how do we use this information to maybe find drugs or therapeutics that could work against the virus? And we use correlation engine to do this again. What you can have is their database has literally thousands upon thousands of studies where they take a specific tissue, they add a drug, and then they get a expression profile. So they've got this huge pharma data in there, pharma list in there. So what we can do is take our signature, which is this is at 1 day post-infection, right? These are the genes that go up and down in our meta-analysis. We throw it in their database, and then what we can do is say, all right, find us chemicals where you either have the same effect or the opposite effect. So in this case, they use this, in this study, used Ruxolitinib. Terrible saying drug names, but so it basically you develop a profile. They have a profile in their database of this compound, right? They go up, they go down. When you compare the two profiles, we find a lot of genes in common, way more than you expect by random chance, right? 2 times 10 to the negative 57th probability that you would get all these genes in common. But that's not the interesting part. It's that the genes that go up in our study or our profiles go down in their drug study and vice versa. If it goes down in their drug study, it goes up in theirs. And the probability of that happening, I mean, this one's 1.4 times 10 to the negative 101. so, so not only are we finding genes, that we're finding compounds that can interact or basically change genes that are associated with our infection, but also we can look for compounds that reverse that. So the thought being, if you can reverse that changes in gene expression brought about by a disease, you have a good chance at actually treating it. And this is just showing you here, again, here's our genes, MX1, it went up in ours, and then in their study, when they did the drug, it went down. So that's the kind of things we're looking for. Here are some of the drugs that we looked at that came up in our screen that were negatively correlated to our profiles. And these are the different profiles at different days post-infection. The higher the score, the better its ability to reverse the changes in drug expression. So I thought this was interesting. Obviously hydroxychloroquine was a very controversial drug. Back when it came out, when COVID came out, it does actually, when you look at it, it seems like at very early stages of the infection, it'll probably does a good job at reversing the changes in gene expression. But as you'll notice, as the infection gets worse, really has no effect. So in a way, they halted this clinical trial of hydroxychloroquine, but they were only treating the really sick individuals. So it's not just a drug, but which phase does it work in? And I would say, this might be preventative, but you should definitely not take it if you're really sick with COVID. Conversely, we see this ruxolitinib right here, which seems to work just about on every phase. And sure enough, people have used that on COVID-19. Again, this is our data from mice. We're seeing that, we're finding this drug seems to work really well. Reverse the changes in mice, probably would do the same thing in humans. And here's just some of the other compounds that we found. Again, in mice, different virus, looking at genes that change, compounds that will reverse that change. And sure enough, everything they're using in clinical trials tends to show up in our screen as well. But obviously some of these, like this estradiol, seems to have a terrible effect. So the more negative it is, the better it is at reversing those changes. So anything positive would seem to be kind of like the disease, more of an inflammation response. So I definitely want to use this, but again, ways to use the gene expression that we looked at to try to find drugs that might actually treat this disease. And I'm really into natural compounds. Again, I'm from Colorado, got the mountain behind me. We're very active here. I'm half hippie, give me something, something without, complications. I want something natural to look at. So we actually did. We went through, looked at some natural compounds. There's profiles in their database. What we found is that cryptoxanthin, and I'll tell you what that's in, but it seems to potentially might be a preventative way, right? Looks at very early stages of the virus, but then you have also this emodin, which seems to work at later stages. It's in the blue here. And again, it wasn't just, there was, as this was going on, I think there's been a big desire, especially in Israel and China, to look for natural compounds that potentially could treat this disease. So again, maybe what you do is, take cryptoxanthin to prevent symptoms, and then emodin, if you do get sick, maybe will help relieve the symptoms. Just really quick, cryptoxanthin is a natural carotenoid pigment. Right here, it looks a lot like beta carotene. Foods that are rich in that, butternut squash. Yeah, I wish it was cheeseburgers and candy or sugar, but no. I'm not a big squash fan, but I do actually eat butternut squash during flu seasons because I think it probably prevents me from getting infected by viruses. And how that works, it's probably a source of vitamin A. What happens is, what we found is that you actually decrease alcohol dehydrogenase. That vitamin A, what you do is you're probably given an alternative route to turn into retinoic acid, which induces all kinds of anti-inflammatory stuff, which regulates the innate immunity and modulates adaptive immunity. So what cryptoxanthin is probably doing is protecting you by making more active vitamin A. Here's emodin, plants that produce emodin, rhubarb. By the way, I have tried aloe, just eating aloe. It is horrible. I don't recommend it. Go get the pills. Don't eat aloe. But what was interesting about emodin is it's been working in lots of different viruses. They show this kind of anti-proliferative effect, or being able to contact viruses. And here, this was in 2007, they found emodin blocks the SARS coronavirus spike protein. Again, this was before COVID-19. They're looking at this. So again, we found this in mice, translated to there. Almost done. So what did we learn? Lots of information to do your own research. Can use related viruses hosts to get insights into another condition. Again, we use mouse and another virus, and it absolutely applies to humans. Same genes that are important in SARS-CoV-2 response are important in the mouse response. If you want to prevent COVID-19 infection, try eating squash and citrus fruits. And maybe if you get infected, try ingesting rhubarb or aloe products. And again, I highly recommend the pills. Probably don't eat aloe. Just acknowledgements here. Again, all this work, there's a paper out there we wrote, but I really want to, none of this would have been possible without Jim Flynn, Mehdi Amadi, Chase McFarland, Michael Kubel, Mark Taylor, Zhang Chen, Enrique Torquia, and they were amazing in helping me put this together. And again, we didn't spend any money. All we did was take other people's data, analyze it, and we got this huge, interesting paper out of the whole thing. So again, you don't need your own data. You can absolutely publish on other people's data. And because this is a graduate thing, somebody sent me this. Us bioinformaticians love Venn diagrams. As you start your careers, here's the ideal. It's to be in the middle here, right? Find what you love, what you're good at, what the world needs, and then, what you can get paid for. And if you stay in this middle here, you're really going to enjoy your career. And I would say, for me, I'm definitely in the middle here. So I'm pretty happy with what I'm doing. But thank you very much. And again, thank you for inviting me to be one of the keynote speakers. And I'll take any questions.